Let's think about the first part of the visual cortex, which is called V1 primary visual cortex. So just to remind you, the retina passes information to the lateral geniculate nucleus, and the lateral geniculate nucleus arrives at V1. Where is V1? Here's our brain, cerebellum, brainstem and spinal cord, very approximately drawn. V1 is at the back here. It's also known as Brodmann's area 17 or primary visual cortex. So there are some things about visual cortex which are worth remembering. So the first thing is that it has 6 layers. In fact, most of the cortex, if you look at the arrangement of it, you find that most of the cortex has 6 layers in different thicknesses. And the main thing to realize here is that the layer 4 is the input layer. So what actually happens is that if you look at the cortex, you find up on the surface, you can identify different layers, 1, 2, 3, 4, 5, and 6. And then underneath this you have basically white matter. So the input from the LGN comes in at layer 4, and remember you've got this double input of magnocellular and parvocellular. And so the magnocellular goes to, so there's if you look at 4 and you expand 4 somewhat, you've got different layers. One of them is layer 4C, and 4C itself is also divided into 2 sublayers, just to make life complicated, more superficial alpha and the deeper beta ones. So these go to the alpha and these beta. And then what happens is that the stream splits in 2 directions, and generally as it goes downwards, this is pathways which then project back downwards towards the LGN, and these project to other cortical areas. And in the cortex, processing takes place in terms of object recognition, mostly in this stream here. So those are the 6 layers of the cortex. We can look at those in a separate video. The other thing to realize is that the cortex has several maps. What are these maps? Well, first of all, there's a retinotopic map, which means that as you look around V1, you can map the retina onto V1. The second one is a ocular dominance map. What that means is that if you're poking around in different parts of V1, so let's expand this bit here, and we've got V1. Each point corresponds to a different point in the retina, so it's a retinotopic map. But you also find that if you map which ones respond to the ipsilateral eye, you find that they form these bands separated by ones that are affected by the contralateral eye. So that gives you these columns in which the cells are driven by either the ipsilateral or the contralateral eye. And these are called ocular dominance columns. The next level at which we have mapping is orientation. What do I mean by that? Well, some cells in V1 are sensitive to the orientation of a stimulus. So if I have an edge or a line like this, and I'm recording from a cell, at one orientation I find not much excitation. If I change the orientation now, I now get more excitation. And so in other words, they have, as I go through all the angles, what I find is that the firing rate has a peak at a particular angle. And this is its preferred orientation. And if I now go back to my V1 and I pick a cell here, say, and I record its orientation, I find maybe its favourite angle is maybe that one. If I look at the cell right next to it, I find that it's got a similar orientation angle but slightly different. If I go to the next one across again, can you see what's happening? We get these columns of preferred orientation which are all arranged next to each other in a row like that. If instead of going to the side I go straight down, so I'm now going, so these are the layers again of the brain, 1, 2, 3, 4, 5 and 6. If I now go in this direction, I find that all the cells have the same orientation. If I now drill down here, right next to it, all these have the same orientation as I go down. Drill down here, all these have the same orientation. So we actually got orientation columns. And these all correspond to the same point on the retina. So in fact, each point on the retina gets represented by several columns. So this here is all one point on the retina. Of course, it wouldn't be that big in that V1, but I can pick out columns all corresponding to that same point, on the single point, same pixel if you like, but representing, not the same pixel, the same area, but representing the whole range of orientations that I might have for that edge. So going back, so then the other thing that we need to think about for V1 is processing. How is it processing information? And the answer to that comes by looking at the receptor fields. We've done that a bit by looking at the edges here, but let's take a little step back and think about where the information comes in to the cortex as we've seen here. What happens is in layer 4, so let's start with layer 4, we find receptive fields that are actually quite similar to retinal ganglion cells. They have a central on and a surrounding off, or conversely, they might have a central off and a surrounding on. So these we call simple cells. You also get simple cells with slightly different receptive fields that look a bit like this, where you have a center, an elongated center, which can be on or off, and then an inhibitory surround on the side, like that. Conversely, you might get off and then an excitatory surround on the outside. These also are simple cells. The next level up in the hierarchy are called complex cells. These have a preference for a particular orientation, so they like an edge of a particular orientation, but they don't really mind where in their receptive field this orientation is. So they're much less position position fussy, if you like. So provided this is in anywhere in its visual field, then this complex cell will respond. If it's outside its visual field, it doesn't respond. Then as you go further up the pile, you have cells with much more complicated properties, where they might be similar to the complex cells, but they don't really have any interest in where in the receptive field they are, where on the retina they are. You also have others which are excited by edges or by bars with the inhibitory surround, and also we start to see specificity for movement in some cells. So as you can see, these are called hypercomplex cells. And what's actually happening here is we're getting more and more abstraction away from simply looking at patterns of contrast at a particular point in a retina, now more towards, so this is more to do just with contrast. Now we're getting more into edges, and then in the hypercomplex cells we're beginning to put it together into more complex edges. And so what we're actually doing here is we are abstracting features. It's feature abstraction. So those are the main features of V1. So one last point that we haven't covered, last issue, is blobs. What this is interfering with this kind of pattern that we've already seen, is we find in the more superficial layers bunches of cells which seem to break up this pattern, and they stain for cytochrome oxidase. And what's odd about these, so they've been called blobs, and they are particularly sensitive to colour. Actually, what I said is not strictly true, and I'm glad I made that mistake, because it's not colour but wavelength, and they're different things. They're sensitive to wavelength, and they are not orientation sensitive. They tend to project to areas of the brain such as V4, which are particularly interested in processing colour. So that's my sketch of the primary visual cortex.